Good morning, friends. Today I am here with Nutrition in Plants Part 2 of 7th Standard. Here in this session we are going to discuss about other modes of nutrition in plants, how nutrients are replenished in the soil and few activities. So let us start the session. First it is about other modes of nutrition in plants. First it is about the parasitic plant. Parasitic plants are those plants which do not have chlorophyll. So they cannot prepare their own food. Then question arises how they can get their food. So they can get their food from other plants. And these plants are called as host. So there is a host-parasite relationship in this parasitic plant. Common example is Cuscuta, also called as Amarville, and it is a total stem parasite. Means this Cuscuta present on the stem of the another plant. Here you can see the Cuscuta. Next is the insectivorous plant. Insectivorous plants are those plants which get their nutrition from insects. These plants do not fulfill their requirement of nutrition by the process of photosynthesis. So, they get their nutrition from insects. Common example is pitcher plant. In case of pitcher plant, the leaves get folded and modified into a pitcher-like structure. So, that is why this plant is called as pitcher plant. And the end of the pitcher is having a lid, and this lid can close and open. So when insect enter into the pitcher, then the lid closes and the insect is then digested by the digestive juice which is present inside the pitcher. Means pitcher contains some digestive juice that is responsible for digestion process. So whatever the nutrient is present in the insects, it get digested by the digestive juices present inside the pitcher. Here you can see the pitcher of pitcher plant. You may here observe that presence of lid, and this lid can close and open. When insect enter into the pitcher, then automatically the lid closes. Here also these are the few insectivorous plants. Now another mode of nutrition in plant is saprotrophs. Saprotrophs means these are those plants which do not have chlorophyll. So again they cannot prepare their own food. Then how they get their food? So they get their food or nutrition from dead and decaying organic matter. And common example are mushroom, bread mold, etc. So this mushroom and bread mold get their nutrition from the dead and decaying organic matter, and these organic matter are of dead and decaying parts of plants and animals. They produces some digestive juices on the dead and decay organic matter and convert them into solution, and again they absorb the nutrient from the solution. Here you can see the mushroom. This is a saprotroph. Here it is about the bread mold. It is also saprotroph. Now next one is about the symbiotic relationship. Some plants live together and share for shelter and nutrients. And the common example of symbiotic relation is lichen. Lichen is just like a couple consisting of one algae and fungus. So they live just like husband and wife. One is preparing food and other is giving shelter as well as helps in absorption of food, nutrients. So here the fungus provides shelter, water and minerals to the algae, and algae provides food to the fungus. So which is prepared by the process of photosynthesis, because algae have chlorophyll, so they can prepare. But fungus do not have chlorophyll, so they do not prepare their own food. So they live symbiotically and getting mutual benefit from each other. So here, in case of this lichen, algae and fungus live together and they are mutually benefited from each other. Here it is the lichen. And there is a very interesting thing about the lichen. Lichens are very good pollution indicator. Lichen only grow in pollution free area. So where there is presence of lichen means there is no pollution. Lichen do not grow in polluted area. Now the next topic is how nutrients are replenished in the soil. Replenished means how they are getting back into the soil. We know that plant absorb nutrient from the soil. So whenever the plant absorb nutrient from the soil, the nutrient in the soil become less. It goes on decreasing. So it is necessary for the replenishment of the nutrient in the soil. So for this, farmer uses nutrients to replenish the nutrients. They uses manures as well as fertilizer to replenish the soil. And again, there is one bacterium that is called as rhizobium. It is present in the root nodules of leguminous plants like gram, peas, beans, etc. And this rhizobium have capability to fix atmospheric nitrogen. Means it convert the atmospheric nitrogen into soluble form of nitrogen, which is further utilized by the plant. So in this way, the nitrogen is replenished into the soil, and nitrogen is very important for the proper growth and development of the plant. And what the rhizobium get from the plant? Plant in turns provide food and shelter to the bacteria. So here also there is a symbiotic relationship between root nodules of leguminous plant as well as rhizobium. In this case, both root nodules of leguminous plants and rhizobium bacteria are mutually benefited from each other. Rhizobium provide source of nitrogen to the plant and plant provide shelter to the bacteria. It is the figure of manure and fertilizer. This is putting manure and fertilizer into the crop field. Now it is about the root nodules of leguminous plants. You can see here the root containing several nodules, and in the nodules rhizobium bacteria is present. Now let us see few activities. First activity is about how that it shows that sunlight is necessary for photosynthesis. In the previous session we have discussed that there are 4 raw material of photosynthesis and one was the sunlight. So whenever any one of the raw material will be not present, then there will be no process of photosynthesis. So this activity indicates that or confirms that or shows that sunlight is necessary for photosynthesis. For that you have to take 2 potted plant of same kind, and then you place one potted plant in sunlight and other in a dark room for 2 to 3 days. After that you take a leaf from both the potted plant and we will test it for a starch. For testing a starch, you put the leaves in a test tube. Now pour some spirit in it and put the test tube in a beaker containing water. Why we are doing this? So that the chlorophyll present in the leaf may come out into the spirit. So for that boiling is must. So boil the water till all the green colors, that is chlorophyll in the leaves, comes out into the spirit. Then what you will do? Take out the leaves from the spirit and wash it in water. And after washing it in water, put both the leaves in a petri dish, or you can also place it on a filter paper and pour iodine solution over both the leaf. Now you will observe that the leaf of the plant which is kept in sunlight will turns blue black. Why it turns blue black? Because here the carbohydrate is prepared by the process of photosynthesis. Why? Because it was kept in sunlight. And the leaf of the plant which was kept in dark does not turns blue black. Why? Because here the process of photosynthesis does not takes place. So this experiment or this activity shows that sunlight is necessary for the process of photosynthesis and thus for the formation of starch. This is the pictorial figure which illustrates this activity. Now next is to observe the fungus bread mold in bread. This activity you can do it in home also. Take a piece of bread and moist it with the help of water and leave it in warm place for 2 to 3 days. You will observe that fluffy patches appear on them, and these patches are nothing but they are the bread mold. You can observe under the magnifying glass or in the lab you can observe under the microscope. You will find that some cotton like threads are appearing or seen on the bread. This is the bread mold, that is rhizopus. This cotton like threads are fungus bread mold, and this is also called as rhizopus. So bread mold is a saprotroph that get the nutrition from the dead and decaying organic matter. Here the bread mold is getting nutrition from the moist bread. So it is saprotroph. In the figure you can see the bread mold. Here also the spores of the bread molds are present. Thank you. For any kind of doubts, please write your question in comment section.